Hi, I'm Nicole, one of the technicians here at Pet Vets, and I'm going to show you guys how we stain slides for cytology. So if the doctor finds a mass on your pet, sometimes they'll poke it with a needle to get some cells out of it and put it on a slide. And then we'll stain the slide with a special type of stain so that the doctor can look at the slide under the microscope and look at the cells and get some more information as to what kind of mass it might be. So I have my slides here. The doctor has poked the mass already. The slides have dried, so we want to make sure the slides are dry. And then I'm going to put it through just these 3 stains here so that the doctor can look at the cells. So the first bin is called the fixer. So all it really does is fix the cells to the slide so that they don't wash off through the process. So I'm going to put them in. I have 4 slides. I'm probably just going to put them in 2 at a time so they're not crowded, so they get a good amount of the stain on them. So each bin has to be about a minute or so in each one, a minute to 30 seconds to a minute. If they're looking for certain cells, they like them stained a little bit darker because those cells have certain things in them that show up better when they're dark. So sometimes we'll agitate them a little bit just to kind of make sure all of the stain is properly making it around the slides and getting affixed to the cells. And then we'll rinse them, let them dry, and then the doctor can look at them under the microscope. So we have just very fancy tools here. So we have just a pair of tweezers that we'll take to move them over. So from the fixer, they go into the first color, which is kind of like a light pink color, so it'll stain certain things a light pink color. And this is the first of the stains, and then I'm going to take my other 2 slides and put them in so that they can fix. This one's not quite dry, so I'm just fanning it a little bit to dry it completely, because if we put it in there when it's wet, everything will wash off. So hopefully once it's dry, things will stick to the slide a little bit better. And inside these little glass jars, there's little slots so that the slides can sit in the slots so that they stand upright. Because if they fall to the side, then we won't have enough of the solution on each slide. So again, I'm just going to jiggle these a little bit just so that way they can have good contact. And the reason why we use these tweezers is because we don't want the stain to get on our hands, because much like it stains the cells on the slide, it'll also stain your fingers. So similar to if you had Easter egg dye at home, it's the same kind of understanding, but this is obviously scientific dye specifically meant for staining slides. So we'll go from this pink solution into the purple, and that's called the counter stain. And this is the one that sometimes they like us to leave the slides in for a little long, so that way it picks up the most color. So then we just move them along, and we keep the stains covered, 1, to keep them clean, and 2, they have an alcohol base to them, so especially this first one, the fixative, will evaporate over time if we don't have it capped. So once I'm done, I'm just going to cap it off so that way we preserve the stain and it stays as fresh and clean as possible. Once the slides are completely stained, we're going to rinse them, and then I'm just going to leave them here to dry. Because when the doctor looks at it under the microscope, they're going to want to put oil on there, so the water and the oil don't really mix very well together. So in order to get the best picture, they're going to dry completely, and then the doctor's going to look at them under the microscope. So this process, the whole process takes, I'd say, about 10 minutes or so. So usually this can be done while you're here waiting with your pet in the clinic so that we can have results. Sometimes the doctor will also call you later with those results so that they have more time to evaluate the slide if they need it. So from here, I don't know if you can see, but if you can see, there's some purple strands left over here. So that's what's going to be left for the doctor to look at. So that's actually the cells on the slide that have stained. So we're just going to let these two dry off to the side. So that's why we have this very lovely colored paper towel here that's been just leftover slide stain as they've been drying. So we put that down too, again, because it will also stain the counter. So we don't want it to stain the counter, we don't want it to stain our fingers. It's not harmful, it just is a little bit unsightly. So we'll put those last two slides in the last stain. And then once those come out, I'll show you those. Those will have lots of nice color on them because they've got the most cytologic material to look at. We'll also do cytology on ears. So same idea. We'll use a cotton tip swab and we'll take some material from the ear, roll it on the slide, and then we'll use this second set of stains for that reason. So the ear material tends to have a little bit more substance to it and definitely potentially bacteria. So we usually try and keep those separate from the cytologies on masses so that we're not getting any cross-contamination. And these stains are also changed fairly regularly to keep them as clean as possible. Alright, so again, so if you look at this slide, you can see all the streaks of the purple there that shows all the cytologic material that the doctor is going to have to look at. So that's it. So these are, again, another slide with all the material for the doctor to look at. And then we're done. So then from here, the doctor will look at the slide under the microscope and be able to talk to the doctor about the results. Thank you.